Hi everyone, welcome back to the channel. This is Dr. Asi, and today's topic is chapter 9, gene to protein. This is also can be called gene expression, like begin gene expression 1. So last time we learned about genes and genetics, especially Mendelian genetics. So now we are going to move to gene expression and talk about genetic code, transcription, translation, and how transcription is DNA directed while translation is RNA directed. Again, this is chapter 9 from your orange book. Okay, let's get started. Okay, so now we have a big summary slide starting with actually student learning outcomes. This chapter is chapter 9, gene to protein. Think about what are the two things you learned past week, you studying the material. And now let's move to this week's chapter, gene to protein. So there is something called central dogma of molecular biology, which you already know. It moves, genetic information moves from DNA to RNA to protein, right? So we already know about DNA part, but today we are going to focus on RNA part. So also we'll talk about gene structure and transcription. Transcription is basically a DNA directed, and it's basically you will learn about genes, promoters, enhancers, introns, exons, coding regions, transcription, initiation, elongation, and termination, and also RNA polymerases and transcription factors. Then the next one, next subtopic is RNA processing, especially in eukaryotic cells. So explore post-transcriptional modifications of RNA. And the next topic is mRNA translation. So, and then we'll talk about how actually structure and function of ribosomes in this topic, and tRNAs and amino acids and initiation, elongation, and termination again, in this case of protein synthesis. And then genetic code. So always remember genetic code is universal. So for all living organisms, that means they exactly use same genetic code and codons to actually convert them proteins. So a few key points here. So here are RNA types. There are more than 8 different types of RNAs. We will talk about 2, 3 of them in this chapter, but there are more than that. Just remember that, and genetic code is 64 codon. You can see this table here inserted inside here. So genes specify proteins via transcription and translation. That's number 1. Number 2, transcription is DNA directed. And number 3, translation is RNA directed. And then number 4, mutations, one or more than one nucleotides can affect protein structure and function. So this is you can see here, this is a nice actually chart which basically summarize the whole chapter 9. You can see here in this concept map. Now, everything is really nicely visualized here in this concept map. You can see this is a eukaryotic gene expression. You can see nucleus in purple and DNA inside here, transcription, making mRNA leaving and going, finding a ribosome, getting inside ribosome using anticodons and tRNAs, and then producing final protein. You can see here eventually at the end of the process of translation. This what you see here is phenotype. Remember our formula: phenotype equals genotype times environment. Genotype is your DNA, but phenotype is your final protein in this case. It's just physical trait which arises from action of these proteins. You can read about this some case studies in your book, Concepts of Biology. So genes specify proteins via transcription and translation. Okay, so now we know that there are 2 big processes. So 2, transcription, translation, right? So 2 stages. This is really started early in 1902 with some experiments we know, which is really creating mutants in different universities, especially Cornell University. You can see George Beadle and Edward Tatum first producing some bread mold mutants using X-rays, and then another Cornell scientist, Adrian Serp, and together with Horowitz, classifying some arginine deficient mutants, and then the hypothesis, one gene, one enzyme hypothesis by George Beadle, and eventually winning the Nobel Prize. Today, actually, this is actually beginning of this whole gene to protein concept. Okay, so now this big concept map, remember, that was for eukaryotic cells, but prokaryotes like bacteria, they can also do, they do transcription translation, since they use exactly the same genetic code, right? The 64 part table, which we call genetic code. So, but they have a little much simpler transcription and translation. Everything happens inside cytoplasm. They don't have a nucleus, but they do have ribosomes. And the second picture is our eukaryotes that we seen in 3 slides ago. So central dogma is going from DNA to mRNA to protein. Now we know information flow from this direction. Now we know that. And so that makes DNA sequence very important. If it is ATCC or ATGC, that makes a difference, which basically it's gonna, each of these triple nucleotides are codons. You can see here, UUU, UC, these are like phenylalanine. AUG is methionine. So you don't need to memorize this whole 64 codons, but you need to remember 4 of them at least. So the whole thing is called genetic code. 4 of them. Number 1 is AUG. So it's methionine. Why AUG important? Because it's start codon. And then if there is start codon, there must be some stop codon. There are 3 of them. UAA, UAG, and UGA. So it's like you are annoying, you are gone, and UGA, you are gone now. So 3 of them, these are like stop codons. Okay, you've gone away. Okay, UGA. So, okay, this is called genetic code. Every organism knows this. We talked about already prokaryotes versus eukaryotes. Transcription is the first part, like the first process, which is DNA directed. Okay, so is made of 3 subparts: initiation, elongation, termination. So in both prokaryotes and eukaryotes, you see there are some other molecules there besides DNA that helps. One is called promoter, which is start the transcribing the sequence, signal the sequence for transcription. It's called promoter. Okay, then we have transcription factors. They also help guide the transcription. And then we have an enzyme called RNA polymerase 2, which also helps. We must have this enzyme. So I think it's better if you see it in a picture. So first, once transcription is transcribed DNA to mRNA, which is messenger RNA, is released, but mRNA is in eukaryotes is processed. So it's processed. So a few things happen to this mRNA. First, it's actually there are these exons and introns. You can see here exon, intron, exon, introns. You can see the introns got cut out. Exons are the parts that they are expressed. So introns gone now, and now this is mature mRNA. Okay, and then 2 things added to the 2 ends. 5' cap added to the 5' part, 3' poly A tail added to the other end. These are three things in processing eukaryotic mRNA. Then the translation is ready. So this mRNA now is more compact and processed and moves out and finds a ribosome. And now it needs to start initiation, elongation, and termination of translation. So we need AUG as a start codon. So, and then the tRNAs, these guys, which bind to anticodons and one end, and then other end, they are bind to an amino acid. So, okay. So it goes and attach them and then start elongating it to the left here. And then amino acids start produced and grow. And then when it starts seeing a stop codon, like we see one of these three stop codons, and it stops when it reaches the stop codon. Okay, so that's translation. Genetic code we already talked about. If you have a DNA sequence like this, as you see in the blue DNA nucleotides, you can see here, we create this red mRNA sequence, and then this purple polypeptide protein with, in this case, four amino acids. You can see here. So I already mentioned that there are other molecules help here. There is one guy here, which is RNA polymerase, which catalyzes. It's you can see an enzyme which catalyzes this whole process of transcription. And RNA polymerase 2 specifically here. Promoter is where actually gives the signal to start. Terminator ends the transcription. And so these are some other things involved here. So you can see here, RNA polymerase is this bubble here. You can the enzyme actually moving slowly. But once the transcription factors actually give the signal, RNA polymerase start actually moving, right? You can see here. So transcription factors giving the signal. And then there is an array here called promoter, which is called Tata box. Let's actually give the green signal. Now, RNA polymerase bubble, it moves, it elongates, and then start making these mRNA, newly made RNA. You can see red color. Okay, so this bubble moves along the DNA and untwists the double helix, 10 to 20 nucleotides at a time. And then nucleotides edit the 3' end of the growing RNA molecule. And then this starts the RNA transcription. And it's terminated once actually and reached to the end. So what happens after that? It can be altered. The ends. We already talked about the mRNA ends, 3' and 5'. Two things added here, and we already talked about it. There's something called RNA splicing and splitting genes. So that's actually getting rid of these introns part. So it's called RNA splicing. Remember, introns are non-coding parts of the DNA there, RNA there. So transcription and translation. Remember, we need a ribosome, and ribosome exists in every cell in every organism, and they use exactly the same genetic code. That makes it very easy for you can maybe produce a plant gene can be produced in a bacterial cell because they use same genetic code. They just need a transfer RNA. It's called tRNA, which are these guys here. They look like adapter. Two ends. You can see here amino acid end and an anticodon end. tRNA look like this, and again, each carries two parts. Here they talks about some other enzymes involved. matching the correct match between tRNA and amino acid. An enzyme involves amino acid tRNA synthetase. So for the correct match. And then once the translation completed and reach a stop codon like UAG, UGA, UAA, and it gets terminated. It's called termination of translation. So the elongation continues until it reaches here. Once it reaches there, then the termination happens, and then basically releases a polypeptide protein, and translation assembly comes apart. You can see here of ribosome. So this is basically the same thing is summarized here. So in this chapter, there is a little information about one other topic here, which is viruses. So last 4 years, we already seen one basically one pandemic of a virus, which happens every 100 years or something. But there were some other ones before this pandemic, and you can see here some examples: Spanish flu, and how many people actually died because of these different pandemics. You can see here, third plague, COVID-19 was the one for the last 4 years. 6.9 million people died because of COVID-19 pandemic, and these are some other ones in different centuries. So a little information about viruses. Basically, the viruses are not really a true living organism. They are just infectious particles with some kind of gene-like things inside or RNA, and they can actually go and infect things. So there are bacteria phages, which is basically they infect bacteria. They can cause diphtheria or some other bacteria-related infections. Here you can see there are plant viruses, which first virus was discovered in plants like TMV, tobacco mosaic virus, and the RNA viruses like the SARS and the COVID-19. And there are animal viruses, so they infect animals, then cause diseases. Of course, flu or influenza, which is flu, is the most well-known one. RNA viruses cause flu, common cold, measles, mumps, and polio. And then the DNA viruses may cause chickenpox, herpes, and stuff like that. You probably already know that everyone going through this testing for viruses, like coronavirus, using a technique called PCR or some other simpler techniques, find out if it is positive or negative. And there are vaccines, and the first time we had a vaccine made from mRNA. So it's all RNA related, RNA virus, mRNA vaccines. So these are some of the last 100 years, some major emerging virus outbreaks, and you can see in this board map, and you can see their names and their years and a little information about each. So that's pretty much chapter 9. This is basically to sum up, just to recap, we talked about gene to protein, so it's also gene expression and a little regulation, so which includes transcription, translation, then the final protein, which is our phenotype, and also briefly talked about viruses in this chapter. So thanks for watching, and please feel free to comment, subscribe, share, and like. So see you in the next video. Happy studying.